Thank you for the opportunity to present today on behalf of Dragonfleye Therapeutics. So at Dragonfleye, we're designing and developing a novel therapeutic platform for a number of indications. And in particular, we want to perfect our access to the subretinal and suprachoroidal spaces in a minimally invasive manner and for in-office use. So this is our prototype device. You can see that it has a red light that transilluminates the sclera, so you can see where you are inside the eye. It has a variable control needle that advances the needle slowly, which accounts for variations in scleral thickness. It also has a motorized infusion so that when you're advancing this needle, and if you're doing a suprachoroidal injection, you can pressurize it so that as soon as it hits the suprachoroidal space, it will automatically flow into that space. Here's a video demonstrating when I was doing this procedure in the porcine model, and we can either use the indirect ophthalmoscope or wide field viewing. And here you'll see internally the red light that we see that we can visualize where we are. And then if you look very closely here, you'll now see the fluid going into the suprachoroidal space in a very elegant manner where the surgeon can see exactly what's going on. Our first indication is retinal detachment because there's a very large unmet need here. Patients have very invasive procedures. They have large gas bubbles. They can't see for many months. They can't fly. They develop cataracts. And we've also done a lot of research demonstrating that the current technique, which is most popular, vitrectomy, attaches the retina in the wrong position. We can see from this autofluorescence, the white line indicates where the vessels were before, and the black lines show you where the vessels are now. So this is why patients don't see as well as they should. So how can we improve retinal detachment repair? We want to avoid the use of large gas tamponades, eliminate these invasive procedures, and be able to do this in the office. So using a pre-prototype device, we've performed this in humans and published this extensively. And this is how the procedure would work with our device. So you would use it like an indenter. You would indent the globe where the retinal tear is. The red light would show you where you are. And then you would advance this needle. And the device can be either foot pedal controlled or with a button on the device. But for retinal detachment, it would be foot pedal controlled. As soon as the needle enters the suprachoroidal space, the bleb would form. This forms a suprachoroidal buckle. And then the retina will attach in a slow and gradual manner, avoiding large gas bubbles, the retinal displacement, and all the other downsides of our current techniques. Here's a patient, a 50-year-old male with early onset Parkinson's disease, retinal detachment, and this was immediately after the bleb was injected, and you can see miraculously on day one the patient's retina is attached, and without any downtime they can go play tennis, they can do whatever they want after the procedure. And here you can see with autofluorescence we don't see those vessel printings, and the retina's been attached in a better anatomic way. Now, what about going beyond retinal detachment repair? In our platform, we have 6 devices that encompass suprachoroidal delivery, drainage devices, and subretinal therapy, all for in-office use. In suprachoroidal, we have the retinal detachment one, as well as the ability to do all types of drug delivery. So what about our drug delivery results to date? We've demonstrated in a porcine inflammation model that we can reduce inflammation delivering 8 milligrams of triamcinolone compared to intravitreal with similar results. And we've also demonstrated the versatility by being able to inject a variety of volumes up to 0.5 ml. And here to give you an indication, you can see 0.5 ml intravitreal versus 0.5 ml suprachoroidal. And you can see how it's a tolerable volume that we are able to inject. We're also assessing the leakage from the hole that the needle makes. And so here you can see the little green area is ICG, which leaks, which is around the site of the injection. But notice at 20 minutes and 25 minutes that there's no extensive leakage. So everything's staying inside the eye despite those large volumes that we're putting in. And here you can see amazingly on the next day how the drug has spread all around the eye in the suprachoroidal space. we're at a stage where we have raised our seed capital of $11 million. We're at first-in-human studies in early 2026. We're continuing R&D efforts for the number of devices that we have, and we're looking to build strategic partnerships with companies that have products that they want to inject in these spaces. Thank you very much.